All right, you're ready.
Okay.
Speak loud.
Good morning, guys. My name is Tino Thoon. This is my presentation on the viability of using the Cas9 system to extend the lifespan of C. elegans at reproductive maturity. Okay, so just to go over some background details, so some of the genes we're working with, one of them is DAF2. In C. elegans, DAF2 is an insulin growth-like factor, and it's the receptor homologue of the IGF-1 receptor, which is found in most organisms that control sort of lifespan and stress responsive organisms. It's found on the third chromosome of the nematode C. elegans. This gene regulates senescence, which is basically aging, the aging process, as well as stress response in the organism. So it, yeah. After embryogenesis and during the development of the organism, all the way up to L2, which is L1 through 4 are the stages of its life, categorized by researchers. It regulates dower formation. Now, a dower is sort of an arrested state in which, if there's limited supplies or resources in the environment, the C. elegans, the nematode, will basically transform into a dower, sort of like a coma, to conserve resources until the environment becomes more hospitable or food is found. Basically, the DAF2 gene operates through the PI3 kinase ATK signaling pathway, working down through those genes to regulate the activity of another gene known by DAF6. Now, just in case you're wondering, PI3 kinase stands for phosphoryl cetidate 4,5 biphosphate kinase. Now, it's another one of the genes along the signaling pathway that also deal with stress response and the aging process in C. elegans. Now, the gene of interest here, or the wonder gene, is the DAF16/FOXO3A genes. They are transcription factors, part of the FOXO4 forkhead family, and they've been shown to significantly prolong the lifespan of mutant animals. Now, when I'm talking about mutant animals, I'm referring to animals whose DAF2, PI3 kinase 8/ATK pathways have been suppressed or completely disabled. So now, when active, the DAF16 FOXO3A transcription factors essentially nullifies oxidative stress in the organism, reversing the effects of damage from oxidation, and it also facilitates increased effectiveness in DNA repair, essentially correcting any errors that occur in DNA copying over many cellular generations. Now, the DAF16 gene codes for the DAF16 protein, which accumulates in the cellular nuclei, which is logical since the DAF16 protein is what reduces oxidative stress and facilitates DNA repair. Since DNA repair happens in the nuclei, accumulation in the nuclei would seem to indicate how it works. Like I said before, the suppression of the DAF2 signaling pathway allows the DAF16 gene to function normally after reproductive maturity. So you might have heard of this system. One of the mechanisms working with is Cas9. Now, Cas9 stands for CRISPR-associated protein 9. It's an RNA-guided DNA endonuclease. An endonuclease is basically an enzyme which cuts DNA, and it's originally found in S. pyrogenes, which is the bacteria that causes strep throat in humans. The CRISPR-Cas system is in a form of acquired immunity in these bacteria, which protects the bacteria against invasion from foreign genetic material, such as plasmids, phages, and viruses. Again, it's a form of acquired immunity, which is usually only found in higher organisms, meaning that it will remember its past invasions. I'm using the word invasions, so that it can protect itself against these sort of incursions later in its life. So what makes the CRISPR-Cas system especially fantastic is that it can identify and accurately cut foreign RNA material or DNA material that is extremely precise, extremely efficient, and no side effects. So it has so because of its ability to identify and destroy foreign DNA in the bacteria so efficiently, it has huge potential for uses in genetic engineering, as you can modify the endonuclease of the Cas system to identify basically to adapt it to any genome and basically use it to edit, disable, interfere, cut, or knock out genes in a genome of any animal, plants, fungi, bacteria, etc. Now this is just a diagram of how the Cas system works in the S pyrogenes. Basically you have a foreign body, usually a phage or a virus. A virus would inject its genetic material. If it's the first time the cell has encountered the virus or the phage or the plasmid, the material, as you can see there, here, is absorbed by the Cas system. The Cas system then reads the genetic material from the foreign body and processes and creates a PAM, or a spacer. PAM stands for protospacer adjacent motif, which it then stores in its own sort of database known as a CRISPR array. Now, the Cas3 system, you can see here, reads the array and creates cRNAs, which the Cas3 system then uses to slice up the foreign invaders like a ninja. So as you can see, it's extremely efficient, and it's very good at doing its job of cutting up foreign DNA in the bacteria. Now, I just want to go over what organism we're using in this research. The C. elegans is a model organism. It's a free-living nematode used for a wide variety of research purposes. Now, the reason it's such easy to work with is because it's commonly found in temperate climates. It's a soil-dwelling nematode roundworm, so that also non-parasitic. And the fact that it's a model organism, meaning that we've sequenced its entire genome, and it's one of the first organisms to have its entire neural net mapped. So we know a lot about this organism. So conducting research is fairly efficient and easy. Now, previous research done in this field of aging and combating stress, one of the first research in this area was done by Professor Cynthia Kenny back in the 90s. She established that the DAF2 PI3 kinase ATK signaling pathway is sort of the pathway that controlled the stress response, development, and aging process of the organism. She also identified that DAF16 and the FOXO3A transcription factors are key for the extension of a healthy lifespan in C. elegans. Now, she found that when you suppress the DAF2 gene in C. elegans, it increased the lifespan of the organism by up to 200% when combined with caloric restriction, meaning a limited intake of calories or energy. However, most of these experiments, the alterations to the genome was done by injecting material into the gonads of adult C. elegans, and then its offspring would inherit sort of these the mutations. So that means that these mutations affected it during development. However, during development, you see that the disruption of the DAF2 pathway caused Dower arrest, meaning normally these C. elegans would only go into Dower form when there was significant danger in the environment or a lack of resources. However, when she disrupted the DAF2 pathway completely before fully developed, the C. elegans, the worm would simply go into Dower arrest and die. Further research, Dr. Ala Ghoshak at Columbia Medical Research established that DAF16 is responsible for the migration of HSNs, which are neural stem cells which originate in the tail of the organism during embryogenesis, but DAF16 during development facilitates the migration of these stem cells to the midsection. However, a hyperactive DAF16 would cause overmigration of these stem cells during development, and under sort of suppression or underworking of this gene would lead to undermigration, leading to problems with the organism during development and premature death. So the methods of the use of the Cas9 system, or for the use of gene knockout or mutagenesis in C. elegans, was established by Professor Daniel Dickinson at UNC, the University of North Carolina, actually quite recently, like 2 weeks ago. So my proposal is because past research, as I stated previously, shown that mutation or suppression of the DAF2 gene signaling pathway during embryogenesis can cause extensive increase in lifespan in C. elegans. However, complications such as Dower arrest and HSN migration errors can yield undesirable results in the organisms, thus leading to problems. My logic is to completely bypass this process because by this lifespan extension, instead of creating changes in the genome of the organism prior to embryogenesis, I propose to change to sort of bring on these mutations in the organism at the reproductive maturity. You bypass the developmental errors completely. Now we can do this because of how effective the Cas9 system is, and it can use a range of different expression vectors such as viral or transfection vectors that can effectively bring on these changes organism-wide, even when it's fully developed. Now, how we're going to conduct this experiment is that we're going to be using the standard Bristol N2 strain of wild C. elegans. This is the standard strain used for medical and lab research. When using C. elegans, this is the same strain used by the previously mentioned studies. Also, we will be acquiring our Cas9 system, which is our Cas9 plasmid, most specifically PDD162, which will be used as the expression vector. We will acquire this from adgene.org, which is a depository database of plasmids and Cas9 systems available for anyone to purchase and use. Now, the mutant DAF2 gene we will acquire, which was established by the Kenyan lab, which will be used as sgRNA or subgenomic mRNA for plasmid construction. Because the PDD162 plasmid doesn't contain sgRNA, or basically sort of like a map to where it's supposed to cut, the DAF2 gene sgRNA will provide that map that the Cas system will need to go in and cut the genes. Now, the temperatures for all the animals in the experiment are to be grown at 20 degrees Celsius, as established by Cynthia Kenyon in 1995. The animals are to be fed E. coli strain OP50, which is the standard food for Bristol N2 strain of C. elegans. Now, we'll be constructing the Cas9 plasmids using the PMB70 gRNA vector, which is used as a method of transfection via microinjection into the organism. We'll be measuring the lifespan starting from L1 or hatching from the egg to or the initial hatching. L1 is the first stage of after the initial embryogenesis development. Death is determined when the organism no longer responds to any pressure or heat stimuli, then we consider it dead. So the expectations of the experiment. If the mutations are successful, we expect a significant increase similar to the increase of lifespan, similar to the results to traditional DAF2 mutants, meaning that even as it's reached its reproductive maturity, it will remain reproductively viable for a significantly longer time than the wild type strains. Now, not from what I could find, there's no established method of attaching any sort of fluorescent or GFP markers to the plasmids for insertion into the C. elegans genome. Thus, I have to rely on the lifespan of the organism to determine whether the mutations were successful or not. However, with any experiment, there are limitations of what can be done. Expression vectors rely on transfection, which is not impervious to the immune system of C. elegans. Thus, not all cell types may be affected by mutations. Actually, in the experiment done by Daniel Dickinson at UNC, only out of 40 organisms microinjected with Cas9 plasmids, only 5 of them had successful mutations. This may obviously leads to inconclusive results that must be verified through sort of later processes. Future research can be done to explore other types of procedures and methods of more stable expression vectors that can affect all cell types, as well as including sort of verification processes such as GFP or other fluorescent proteins.
There's no mechanism to check to see if the gene was taken up. There's no other thing.
I couldn't find any methods where they attach GFP, a fluorescent protein, to the Cas system so that they can be injected at the same time. If I don't do that, I have to do that separately. And then there's a lot.
But separately doesn't work.
Yeah.
You know that they're both uptaken at the same time.
Yeah.
We got to figure out a way to do that. I mean,
Also,
if we do it together, it just makes the experiment a lot more viable. Just go back a few slides. Sorry, I'm taking up time just because there's no more time left. Go back one more. Back one more, right here. All right, so your Cas9 system is PDD162, right?
Yeah.
And then we're going to get the DAF2 mutant gene in a plasmid. What's the process of uniting those two together?
Is
it as simply as combine them in another test tube with
that? From what I could read from the methods established by Daniel Dickinson, that's basically what they did. But they also use PCR to multiply the expression vectors because you don't get a large sample when you buy it.
Right.
So you have to use PCR to amplify.
But we'd amplify it at that level. We'd also amplify
the finished product.
Right, the finished product. Well, you might want to amplify all of them.
Yeah.
Now, how are we actually getting it into the C. elegans? How is that work?
Microinjection.
So how's that work? What's microinjection?
It's like a very small needle and you just inject it into the system.
Are we specifically injecting into the gonads of the C.
No,
elegans,
we're
or just
injecting
into the C. elegans
into
itself?
the worm itself, into the midsection.
Okay, and what stage of its life cycle are we doing it? So actually, let me rephrase that. So here's my question. My question is, are we looking for at the life cycle of that cell, that organism we injected?
Yeah.
Or are we looking to see if that integrates into its genome, and we're looking at the lifespan of the next generation?
We're looking if we can increase the lifespan of already mature adults, so that you don't have to wait till the next generation. You can instill a change of mutation in the current generation.
Why did you choose that PDD162?
That's actually the empty vector strain used by Daniel Dickinson, and that doesn't have already attached sgRNA, which is sort of like the roadmap it needs to identify the genes to cut. So we can attach any gene we want to it.
Okay, so we take that PDD162, we attach the information, the gene of interest, to that Cas9 system.
Yes.
So now it knows where to target,
Yeah.
and then we attach the mutant DAF2.
Yeah.
Sorry. Is it? All right, see you later. And then you attach, so you have to attach two things to it.
And then you
in order to get the,
and then you have to put it inside the PMB70, which is the vector, to sort of get it inside.
Can I get a pass?
Yeah, definitely. Where are you going?
Barry. He's always, I think he lowers the grade every time.
Just tell him to zoom. What class are you in?
I have to.
Go around the back.
So my only thing is, so in order to do this though,
I talked to them this before. I hit a grade.
So we're already classroom. That's nothing. That's cheap. Other than that, what do we need? We need a TCR. I think that's the realm. Really getting out of the TCR machine. Let's do whatever we want. I'm willing to spend. Nobody's else has come up with an idea yet, so until we give you money, feel free to steal all of that $4,500. And the other question is, which we got to figure out.
So like
In a high school setting, are we breaking any laws or any rules by doing this type of work? I don't think so.
I can check like.
But that's what we need to figure out. Is like, listen, is it okay? Like if they do this they probably, I'm sure they work, just look to see if anybody bring a cat in order with the CRISPR Cas9 in a high school setting.
I mean, they sell it on Aggie.org.
Yeah, we can do this at home. I'm saying, in a high school setting, I'm totally down for like, listen, if we're a gray line, we'll work in the gray line, the gray area. I'm okay with being a little bit on the edge.
Also, like L2, so L2
You don't want to check.
I like the article I was talking about.
Okay, so what we need to do is, there's definitely some things we can change, just like you need more images, things like that to kind of follow in your systems. So in terms of presentation, that's just presentation. What you really need to do is, let's make a list of what do we need to change. And let's try to put values to it, so that you can map, and let's get that together. Alright, cool. Good job. Alright, everybody. Alright, so your job, your task for the day is this.
April 6th. April 6th. January 18th.
How old are you?
February 7th.
June 9th.
Good job, Tina.
August 24th. What? 24th.
January.